So we are going to start, and we are super happy to welcome Thérèse Malliavin, who came from Nancy, to present her work on like protein structures, etc.
Okay, thank you for the introduction, John. So, okay, I am working at the University of Lorraine since 2022. But before, I've been spending 15 years in the lab. You know, because you have been inviting Vincent Mallet some time ago. Going from the okay, he was making the PhD between the unit of Michel Nilges and the unit of Jean-Philippe Vert. And so I know quite a bit Vincent because he was finishing the PhD when I was then still in the lab. So okay, just to connect me a bit to the people. I'm sorry for Grenoble, actually, it should be Paris, but I'm sorry for I was delocalizing the seminar in Grenoble. So today I'm going to talk, okay, I am working in bioinformatics, even in proteomics, or structural bioinformatics. So the point is I am interesting myself to the structure of the protein, and the structure of the protein are data, and there are a lot of data attached to the structure of the protein, and then computer science and statistics, and also apply mathematics and provide tools for analyzing, for calculating energy, of calculating what we are calling the conformational landscape of the protein, which can be considered as the free energy surface of the protein. And so that's the reason for which I wanted, and some years ago I started a collaboration with mathematicians working using the distance geometry problem to try to provide conformation of disordered protein. And I will talk about that today. But first I will make some introduction, so it's like that, yeah, about the proteins, because I think it's better because maybe you are not all familiar with the protein. And also I would like to say, if you want to ask questions, please ask, because then it's producing a nice, okay, I hope a nice exchange between the room, even though these things can be then cut when the video is on internet. But okay, yeah, I mean something like. Okay, just to remind you what is a protein. Protein is a polymer of amino acids. So each bit here is an amino acid, and the amino acids are all different because they have a backbone, which is the backbone is this part actually, which is the same all along the protein. So it's a bit this long tail like that. Long wire, and then you have different arcs which are here, which are the side chain of the amino acid, and they are all different. You have all different sorts of them, and they are making different kind of interaction for protein, which are called folded protein, which have some stable structure. Okay, but today we are going more to talk about disordered protein, and I will concentrate mainly on what is called the backbone, actually this part. So for the protein structure, there are different levels of structure. You can define a protein as with a long word, actually, which is a word with letter. So the letter can be three. It's like a big set of words, let's say like that, a big sentence. So it's called the primary structure, and the sentence can be a form of one-letter word or of three-letter words, and there is a way to pass from one to the other. There's no problem with that. So that's the first level of a protein structure. And then you have a second level. When I was showing you the backbone, actually the backbone it can make some what is called the hydrogen bond, which are written here with these small dotted lines, and okay, which are permitting interaction between oxygen and NH groups. Because the NH groups are a bit more positive, the oxygen a bit more negative, so they like to interact a bit some electrostatic way. And this hydrogen bond are making some kind of glue, and you can have some different kind of secondary structure which are formed with this glue. So one which is helical, which is called the alpha helices, and others which are extended. So you have two extended regions like that, which are attached by hydrogen bonds, which are between residues which are far apart in the sequence.
You mean that the bone is evolved vertically and horizontally?
Okay, the fact that it's horizontally or vertically, it's no.
No, but I mean there is the bone part on the vertical of the horizontal dotted line.
I mean, the hydrogen bonds, they are vertical in both cases, but it's just because the figure was put like that.
Yes, sure, but the on the horizontal dotted line.
Yeah, on the backbone, you mean? Oops, yeah? Or the backbone. J'arrive pas à faire ce truc. On the backbone?
Yeah.
Yeah.
So, okay, the backbone is here. It's not the vertical legs.
So the vertical lines are the hydrogen bond between a residue in the backbone, and in the case of the alpha hélice, oops, je trouve un moyen de ne pas faire bouger ça. In the case of the alpha helix, there are residues which are close in the sequence, which are producing the alpha helix, so it's a bit local, some structure which is forming by local interaction on the point of view of the sequence, but beta sheets, which are formed of beta strand, which is the other structure, is formed by non-local interaction. In the non-local, it is because the residues are far apart in the sequence. So that's a bit the difference between the two types of secondary structure. And then you have other structures which are called the loops, which are a bit like that or like that. No, mainly like that, which are a bit attaching the beta strand and the alpha helices. And that's producing the tertiary structure that you can plot like with all the atoms here. Or here we are plotting it just with the secondary structure, so it's a bit more clear. Here you have all the alpha helices around, and here you have the beta sheet yellow in the middle. And you see here you don't see anything when you have all the atoms, but when you have the secondary structure you see a bit better. You can also describe it showing the surface of the protein. And then you have the quaternary structure, which is actually when you have several proteins which are interacting together. So one famous case is the hemoglobin. Which is very famous in the literature. It was one of the first crystallographic structure determined in the 50s. And here is another case, which is the PDZ, which is the PDZ tandem PDZ protein. I will discuss a bit it later. So, but all the protein have all this structure because most of the protein interact with other proteins. So the quaternary structure, all this level of structure are always present in the protein. Okay, see, if you look at the protein like a geometric object, you can define the position of the atom as the relative coordinate, but there is another way to define it, is to define it through, so here you have again the backbone, you can define the relative position using what is called dihedral angle. So the dihedral angle, when you have four atoms, if I'm taking the C, the N, the C alpha, and the C, for example, so you have four atoms. So here is one residue of the protein, and that's the next residue. So you have a repetition of the atom, but there are different residues along the sequence because it's a polymer. And then the three first atoms define a plane, and the two last, so here this one, and the two last atoms are defining another plane, and you can get the angle between the two planes. It is what is called the dihedral angle. And there is a relationship between the dihedral angle and the distance between the atoms. And it is this relationship which my colleague mathematicians they were using to develop the algorithms they developed to enumerate the conformation of the protein. So they are saying they are using distance geometry, but the take-home message here is distance geometry and angular distance and angle are closely related, of course. And angle geometry is more called in the structural biology the torsion angle space, and distance geometry is considered to be very different. But from mathematical point of view, they are equivalent. So it's not very interesting what I'm telling you because you already know that. But it's important to tell it from the point of view of structural biology. And then actually, if you look, you have here other angle, I will not talk about them today, and you have also the omega angle, but which I will not talk, I will talk mainly about the phi and the psi angle. And actually there is something which is called the Ramachandran diagram, which is defining the value of the phi with respect to the value of the psi, so you can put all for a given protein, you calculate all the phi and the psi, and then you are having some points which are defining you the position of the residue on the Ramachandran diagram. And that's the reason here you have the famous alpha helix I was talking here in red, and the beta strand is in blue, maybe a bit also brown here. But Actually, there are a lot of definitions here. I took especially a definition where you have a lot of different regions which are relatively small. And I'm doing like that because it's helping me then to enumerate the conformation, because I don't have too big space in the angle with respect to the angle value. I don't have a too big box to sample. So it's easier to try to systematically enumerate.
Can I ask?
Yeah.
Yeah, about this diagram, because it looks like it's pixelized. Like there is only you take only integral values for the angles, or how?
Okay, here it's plot as pixelized. Actually, here it's pixelized about, so you have, I think, variation of 10 degrees. And when you have variation of 10 degrees, we consider we are about at the same value. And we are not so precise, depending of the structure, depending what you are doing. But what I'm doing here, which will be calculating structure of disordered protein with using nuclear magnetic resonance data, I'm not so precise that I have to know. But I could have a larger amount of points. But what is important here is actually the region which are colored a different way. And this region were obtained by looking at high-resolution crystallographic structure about 15 years ago, and then doing some clustering of this structure, and then finding that there was here you have 10 possible regions when you can put you can have the residue. So here the blue region actually it's connected, of course, because you have something which is a torus.
Okay, so because when I saw this picture, I thought that you had taken one protein and then you were displaying basically a two-dimensional histogram of the angles, but actually it's not what we are seeing. It's more like a map in nature that you never observe things in the white region, and most of the time you fall in those different areas of the map.
That's more something that's not coming from. I would have one protein, I could take one protein and put some across where the residual protein would be. But here it's more a knowledge coming from. So here it's knowledge coming from a large set of high resolution crystallographic structure. So that's giving you this kind of. Oui, yeah.
I also didn't really understand the difference between the angle phi and psi. Why are they different?
The angle phi and psi, actually it's a definition. So the phi is the angle between C and C alpha C, and psi is between N, C alpha, C N. You see, you have, it is the set of atoms. Here I explain how phi was calculated using this set of atoms, and you have the same angle psi using the atom from N to N, actually, the next N. And actually it's the same N, but actually it should be Ni and Ni plus 1, if I would be really very mathematical. Because here you have the residue I, which is here, and the residue I minus 1 is here, but it's not continuing, it's continuing, but I did not make the picture for minus I. And here you have the residue I plus 1. So you have the residue I minus 1, I, and I plus 1. And when I'm putting C here, is C I minus 1, and I, C alpha I, and Ci, if you want to see I being the residue index. Yeah, it's, yeah.
Just the color represents a specific location in the protein, the color of the diagram.
The diagram represents a specific region of phi and psi. And for example, when it's completely red, it's corresponding on the alpha helix, which is here, a pure alpha helices. And then if you have here the green region around is more so-called the 310 helix, which is an helix which is a bit more distorted actually. And then you have another region which is here, which is the polyproline region, when you have even more extended helix. So it's a helix.
But this region may overlap, or
what do you mean
when
by?
You say this region corresponds to?
They are not overlapping from the point of view of the phi and psi.
I mean, the interpretation of the regions. You say the red area is the helix, because it never goes out, and the green area is something else.
No, I'm not saying that they're never going out. I'm saying, okay, what can I say? You are taking a residue in a protein, and you can measure the phi and the psi angle of this residue if you know the sequence. And then you can position the residue somewhere on the Ramachandran diagram. That's one thing. Or you can take your residue, and you don't know the phi and psi, but you know it will be in one of the 10 regions. And the 10 region giving you some bound for the phi and psi. But it's nevertheless there's a lot of possibility which are left. But I don't know whether I answer. Yeah, it's okay. Okay, so then there are a lot of people trying to work on working with protein structure since I would say 50 years, and there is a very important notion in proteins for protein structure determination. Is actually you have different kind of data you can measure with the to determine the structure. You can do crystallography, you can do nuclear magnetic resonance, you can do cryo-EM even. And the idea is that you are trying to obtain a convergence. So you are having some restraint, you are trying to optimize your conformation, and you are doing that several times. Several times it is for the NMR is doing several calculations for the nuclear magnetic resonance calculation. For the cryo-EM, several times is looking at a lot of different images and looking how they are from different angles and so on, and try to get some convergence on the images, and then you get finally go up to the atomic resolution. For crystallography is more inherent to the method, because crystallography you get an average signal somehow. It's average because you have the crystal packing, so you are getting a signal which is already average. And people are happy when they have been able to converge to something, because if they are not converging, they don't know what they have. But there is a problem. For example, here I've been showing I've been showing some ensemble of conformation. Actually, here you don't have convergence because you are missing some restraint at the beginning. So here you have been converging because it's a bit superimposed. And here you have a spaghetti meal because it's not converging because it's a more disordered region. So I don't know, these things is more, and now I would like just to explain how the people are usually doing determining predicting protein structure from the sequence, because it is a goal the people have since some years, and actually it's coming as soon as they started to have a database of structure. There have been people so it was at the beginning of the 90s. They start to look at the few structures that were in the PDB, the Protein Data Bank, which is the big database for protein structure, and they realize when you have the protein, the sequence of the protein are very similar. So that's the percentage of similarity of sequence. So 100 they are the sequence which are the same, and up to 20% they are very different. So you have 20% of identity between two sequences, primary sequence. And then you realize that when the sequences are the same, the structure, the coordinate are, the deviation of coordinate between the structure is very small actually. You have two structures of two proteins who have the same sequence, they have the same structure if they are not disordered. And using that observation, then it's possible to say, okay, here you have 3D, so which is a sequence for which I know the structure, and sec is a new sequence for which I don't know the structure, but then I can use the already known structure to say, okay, here I have some proximity in the space. So I have my green sequence, which is in green here, and then I have the proximity in the space, which is coming from the already determined structure, which are in red. And then you are optimizing, and you get a model actually. And it was okay. There are usually simultaneous method to get some homology model, and that was the first step for predicting a new protein structure. Well, I'm not going. And now the more or less step the procedure everybody is using is actually you are taking one protein sequence, then you can get local information on the conformation, either by looking at library, either by looking at measurement, for example, by NMR, chemical shifts measurement, that we'll introduce later. And here, you have a multiple sequence alignment, so you have been different sequence you have been aligning, and that is giving you, so you look how the residue are getting mutation on the same position, and when they are mutating together, it's telling you that they are close in space, So they can be far apart in the sequence, but close in space, and in that way, you can reconstruct the structure. And actually, it's a procedure for which there was the Nobel Prize. I know that there will be a Nobel Prize when I ask accepted to talk. So that was the Nobel Prize was given last week for this kind of procedure.
And to be clear, for instance, whenever you look at the helixes or the sheets, how rigid are those substructures?
That's a very good question. Actually, the helices, they can be kinked. They have some kink. And also the beta strand can be twisted actually. So and that can be and actually all of that is coming for the hélice. Yeah, but also for the beta sheet certainly it's coming from the variation of this angle omega. I was considering, as I will not talk about it, but this omega also has a lot of importance from the geometry of the product.
And also another question is, what do you call alignment exactly?
Alignment, actually, then you are taking the words which are formed by the sequence, and you are trying to look at the place which are looking similar.
Okay.
And that's usually is done by linear programming, and it's called genomics. Okay, I don't know. I don't want to say something stupid. So I prefer not.
No, I understand, but it means that if you have two proteins with like two patterns in the sequence, but in one protein they are closer and in the other one they are far away, so you align this way.
Yeah, so then you put some insertion and some deletion. So there is a whole bestiaire, a whole zoo of different methods how to deal with the insertion and the deletion. But yeah, then you manage to have a very, very big word. Sometimes you have nothing in front of one sequence, but you have something with another sequence.
But the way they fold may depend on the fact that they are far away or not.
Yeah, alors, so yeah, but okay. What's happening probably when you have a protein, you also can have several domains, and then you can imagine that sometimes you have one domain which is missing, so then the rest is folding as it should be, and then
locally it should.
Yeah, you may have so domain means that they are. We will see one example later. Domain means things which can be a bit independently, even independently studied experimentally, and then. Of course, if you have a short protein and you have an alignment with a lot of insertion and deletion, it doesn't have a lot of value. Yeah, the people are trying to reduce the deletion and the insertion. That's for sure. Okay, so then you have a problem with this approach when you have not so many alignments. For example, here it's an example from a colleague of mine, Alexander Breden. And he has been studying different calreticulin, which are three cases of calreticulin, which are coming because there have been a frame shift. So some of the regions are not expressed actually, are not transcribted in RNA actually. So they are because the protein are coming from DNA, the DNA is transcribed in RNA, and then the RNA is translated into protein. And you may have sometimes some part of the DNA which is not transcribed to RNA, and it can come also by because you have some disease or some it can produce some trouble. And here, what they have been here they have been testing several approaches. So it's a paper already recent, and they have they don't have a good confidence index when they are using AlphaFold or other method, which are the Robeta is the one from David Baker, and because it's one of the programs of David Baker, and they have to decide. They know they have an alpha helix. But around they may have different things, special things which are regions which are disordered, and they have then to rely on more modeling method, looking at the energy of the different system and so on. They cannot use as it the method for prediction. Okay, so a lot of trouble are coming actually from this disordered protein, which are On the same side, they are very important for the biology because they can help the different proteins to interact because they can, to the definition of the intrinsically disordered protein is a protein which doesn't have one defined protein, one defined structure. It can populate many different structures. And then it allows it to interact with many different targets. For example, this is the case for this protein which is called P53, which has two domains, and here you have in a dot dash you have a disordered domain. You have here some conformation, possible conformation, and then the different region, with the red region and the yellow region, interact with two different protein. And this protein P53, it was discovered because it was one of the most mutated protein in case of cancer. And they did not understand where it would come from up to the time they realized there is this disordered region and it has a lot of partner of interaction in the cell. And so if you are touching something, all kind of different interaction can be perturbed, and then you have a trouble. Okay, so here that's another limitation. Okay, that's an example of prediction in a region when you have two domains which are known, and a very big disordered region which are in orange. And AlphaFold is telling you is that you don't have a good score actually. The error is expected to be very big on this region. So you have all these white region, white parts on the diagram, but you cannot say much more than that. So okay, so now I'm going to discuss. So yeah, I already talked 30 minutes, but I'm going to discuss to show you a method that has been introduced by my colleague coming from Leo Liberty is more a computer scientist, and Anthony Cohen is an applied mathematician as well as Carly Labor, and they have been proposing a problem which is called the molecular distance geometry problem, which is like a distance geometry problem. So knowing the distance between point, finding the point, the coordinate of the point in the space, but the points are atoms in the molecule or in the protein. They have been studying a lot of the protein. And studying the protein, they decide to make an order of the protein, which is here shown by this label. So first putting this atom, then next one, and so on, and drawing the protein that way. And as I told you, there is this torsion angle, which are actually dihedral angle, which are concerned with 4 different atoms. And they are related to the distance between these atoms. And in that way, you can, if you have 3 atoms which are positioned, you can find the position of the 4th atom. And you have 2 positions because you have a cosine in the middle, and the cosine it can be, the angle can be positive and negative. So that's the situation you have when you know the distance, they are all going, so you know them precisely. And you can do something also if one distance is an interval, which is in magenta, because you are discretizing the distance. So that way you can reconstruct a structure, and that's the advantage. You can enumerate it. You can build a tree actually, and then you are at the first time you position the 3 first atoms. And then you position the 4th, and then atom which can have 2 positions, and the 5th and the 6th, and so on. Or if you have this, you don't know all the distance. Actually, in place of having 2 branches at a time, you will have 2n branches. So at each level actually. And that looks okay, because like that, in principle, you can enumerate all solutions, but you have a problem which is the complexity of the problem. You have an exponential complexity, and you cannot enumerate all the solutions because it's taking too much time. Okay, so then what I've been trying to do is to take this approach and to try to apply it to the case of the disordered protein conformation. And I was doing that in the frame of nuclear magnetic resonance, which is actually, we can measure something which is called the chemical shift. I will not describe too much what it is, we can discuss that later if you want. And then this chemical shift is giving you an idea of the phi and the psi torsion angle. And it's even so good that you can have you can have neural network to do it. This reference is describing a neural network which has been trained on folded protein. But okay, so there is a relation between the two, and there are also a program to predict the torsion angle for disordered protein, which are less precise and so on. But which is called delta 2D, from delta 2D. And looking at this program, actually looking at the Ramachandran region I've been describing you before, and then doing some calculation on triplet of Ramachandran region. So for taking three protein residues, so you have X, Y, and Z, and each one is in one region of the Ramachandran, for example, A, B, and Z, for example, which are A are the red one, and the D is the green one. Then it's possible to looking at the interval of angle phi and psi. It's possible to enumerate the whole tree for these small triplets, and you can do that also for all the possible regions of the Ramachandran diagram. And then you can look at the variation of the population, the relative population, saying the problem for which you have the biggest amount of solution will be 100%, and then you make the ratio for the others. And in that way, you can then decide that, okay, there are some regions which are more populated than others. So I will decide that for triplet, it's mostly these regions which are populated. So in that way, you are reducing a lot your complexity. You are having less smaller complexity, and then you can rely on the method on which you are taking your whole sequence and you are cutting it in fragments. And for this fragment, you get the phi and psi input for taking the chemical shift, some prediction, then district with the triplets. And then you are running a calculation when you are trying to explore the whole tree. So for each fragment, you have small trees. For each fragment, you have a lot of different small trees, which are the green one. And then you are exploring all these fragments, launching the calculation with a Slurm array on the HPC. Yeah?
And just because probably I missed a step, when you say triplets, it's like triplets of consecutive angles?
Yeah, it's triplet of consecutive angles. It's triplet when you have three residues which are close in the sequence. And I decided I was not taking into account the side chain of the residue. That could be also the next step to do. But I was making taking all the possibility, and for this triplet, looking each time exploring all the small, because the three are quite small, and then looking at exploring with the same level of discretization, then to be able to have something which is comparable, and then looking at the one which are producing the most solution, which is a bit a way to see whether okay. For example, if you have the Ramachandran diagram, usually they don't like much to jump from one region to another and then to come back to the same region. These things are not are usually pruning everything from a geometrical point of view. And then you are having so you are producing conformation from the fragment, and then the idea is that you can attach the fragment by superimposing the two extremity of the two successive fragments, and in that way you get so it's called the hierarchical assembly of fragment, and in that way you get all the solution. But that's working only because you one has to cluster the fragment at each step. Otherwise, you have too many solutions, and you get back to the exponential complexity I've been describing before. And maybe I will jump these things because maybe we can discuss that. That's a big. Okay, I have a way to cluster. If you are interested, we can discuss about that. And so here, actually, I'm not the only person working with IDP. So IDP is the acronym for Intrinsically Disordered Protein. There are a lot of people working on it, but most of the people are using Monte Carlo method to generate a lot of conformation. And then TIDP is using systematic enumeration. And then the next step, when you have the conformation, is to try to see whether they are verifying the ensemble of all the measurements you can do on the protein. And in that way, I'm doing more or less the same thing than the others. I think I will present some few examples of how the TIDP worked, just to show you how it works. And then I will concentrate a bit on this part, because here we have been proposing a method called Ramamix, which is a Gaussian mixture, and maybe I think it is that region, that part, that we could imagine to have some interaction with the method used in shape analysis. Okay, here is just to show you one example of a protein, which is, I was telling you, it was a PDZ tandem. So it's a protein which has, maybe the best is to put it like that. It's a protein on which you have two domains, which are orange and green, with P1 and P2, which actually you see them in orange and green, and they are folded. They are known, it's a PDZ domain, it's a very famous domain, everybody knows how to, you have plenty of them, and it's very well studied, very well characterized. And in between, you have something which is called an EDR for intrinsically disordered region, but it's a bit like a kind of IDP, but attaching the two domains. And using TIBP, it's possible to enumerate the conformation of all the fragments, then to assemble the fragments, and then to get different conformation. And then it's possible to fit the Sachs curve and also different other measurements. And then you get a description of what's going on. So here you say that you have 10% of the population, which is because you are getting the population from the fitting of the one method, which is called the Sachs curve. And you are saying you have 10% of the conformation for which the two domains are close to each other. Then you have 8% which are about 8% for which the two domains are far apart. And then 3% for which are intermediate. And then most of the conformation actually are represented by one conformation for which the two domain PDZ are not close, but not very far apart. They are in the middle. Okay, so that's one example that what you can obtain with this kind of method. So then I want to come back to the filtering of the conformation after having producing them, and in particular the filtering by fitting the Sachs curves. And what I'm using, it's actually a Bayesian approach on which there are two terms. One term is the chi-square, and the other term is called the entropy, which is described by a Kullback-Leibler divergence. And then you have a weight for the entropy, and you are making the calculation for different weights and get a kind of elbow curve. And then when you are trying to get the weight for which the chi-square is not is decent, is not too far apart, because you don't like to be far apart on the data, but at the same time you don't have to. You want to have also to minimize to maximize the entropy, because you don't want to force the system to be in one solution. And actually, I've been using it here. That's the result obtained for, you have three different types of results here. We obtained for three times the curve which are measured on one protein, and I was using the same data set of conformation. And here you have the number of conformations which have been selected by this method, BON, which was developed in the lab of Gerhard Hümer. That's the paper which is describing the method. And you see actually that taking the same set of conformation and three different Sachs curves, you get the number selected, you get 117 is selected each time. you get each time 159 is selected. 159 is selected. I don't see it. I think it's selected here and also 3 times also. And each time with a population which is between 30 and 20. So actually, it is something which is good to filter the conformation and have an idea of the population. But we wanted to propose something else, and then I had a collaboration with my colleague Jérôme Midier, who is here, and for which we were trying to do it directly from the phi and psi value, because actually at the beginning, by looking at the chemical shift, so the NMR data, the nuclear magnetic resonance data, we get some region on phi and psi. We know that they are for which the conformation are populating this region, and at the end we have a set of conformation for which we can describe using normal mode analysis along the internal torsion angle. You can describe them with a harmonic potential surface with some covariance matrix, and that gives you some Gaussian distribution of population. And then what we were doing with Daniel Foster, was a postdoc with me, and Jérôme Midier, we were optimizing the Kullback-Leibler divergence between the distribution of probability, which is on one side the sum of the different Gaussian weighted Gaussian of each conformation, and on the other side the population coming from the prediction from the chemical shifts. And actually we have I was showing the Ramachandran diagram 2D flat image, but actually we are on the torus because the angle are circular, yeah. And then one has to make some change of variable with von Mises distribution, which is here. The expression is for the probability is expressed like that. And okay, that was developed by Jérôme, so I'm not giving. And then to optimize, we have to get the derivative with a different variable of the von Mises distribution, and then we can use an algorithm which this library to solve this nonlinear optimization problem. And that was interesting because actually it was giving us, we were applying to a couple of proteins. And it was interesting because we could we finally found on this protein, but okay, then we are trying to see with what we are doing with marine, we are trying to reproduce it maybe on a larger set of proteins. It's something we want to do during the next month. And if you are taking the conformation, just the coordinates, they are very different. And even you are selecting some using this Gaussian mixture, you have different coordinates. But then, if you look at another parameter, which is I call the local, the profile of local gyration radius. So actually, you get here, you have the sequence, you have your backbone, and then you take a window centered on one residue, and then on this window, you are calculating the gyration radius, which is actually the radius of gyration, sorry, which is actually the square root of the quadratic average of the distance between the center residue and all the residue which are in the window. And then if you have small local radius of gyration, it means that locally your structure is a bit more packed, and if you are large local, it's a bit more extended. And here you have some calculation, some comparison of profile, which here you have the residue number, and some profile you are obtaining. So one is black and the other is red, and they are coming from two independent runs. But then finally, after having filtered them, you get somehow to a kind of converged situation, even though it's not a convergence of the coordinate. It's more a convergence of something which is a local description of what's going on, with something a bit more packed, a bit more extended. That was also we could. We were doing that for the PON, also filter, and also for Raman filter. So that's even able to calculate some kind of matrix measuring the distance between the profile and having a kind of threshold saying, okay, if it's more than 8, okay, we have probably the similar profile and so on. So it was, okay, I'm not going to describe too much that. Here is another example with some colleague from Taiwan, for which I have another protein which is formed of 2 helices, one which is well formed and the other is not so well formed. And then at the beginning, when you do the ABP, you have and can plot the angle between the helix axis and the radius of gyration in function of the angle of the helix axis. And actually, before filtering with BON and Ramix, you get some distribution which are a bit extended around the whole space. And here, it's a bit, okay, I would say it's a bit a slight signal, but you get something which is a bit more like that. You have some shape like that, some bell shape, which are corresponding to the conformation we have been filtering. So it's also some kind of distribution, of convergence. And okay, so just the last, so I would be very interested to discuss with you about that, and that's my colleague. So I have a collaboration with several Taiwanese colleagues, and Bradley Worley was the person who has been writing the first program for doing IVP, which was working really well. And with Daniel and Jerome, we developed the Gaussian mixture, and here are my colleague mathematician. And I'm calculating at the Mesocenter in the University of Lorraine, and I thank you for your passionate attention.
So maybe I can start with a question. Actually, that for me, the point at which I stopped understanding everything was.
I hope it wasn't too long.
No. But it's because okay, I understand that you have your sequence, and then you say okay, when you have triplets, then of angles, you can kind of reconstruct the 3D structure from the chain. And then obviously, if you sample all configurations, it's exponential, so you don't want to do that. And so you have a pruning method.
The pruning method is helping a bit. Yeah.
That tells you, okay, you shouldn't go in this direction. And then at the end, probably you have a prior that tells you, okay, this configuration, it's garbage, you throw it away. So you only explore, it's like rejection sampling for your plausible configuration space, but I didn't understand how you do that.
Concretely.
Yes.
Concretely, okay, we are measuring the chemical shifts. So that's a number which are given by the people who did the experiment.
And chemical shifts is what?
The chemical shift is this NMR data I did not describe too much because actually I realized I tried to make a big introduction, but then I realized if I am explaining, then I am there and I'm staying 2 hours. So I decided maybe we can come back on that. So it is this value, but actually it's not the most important thing, the chemical shift. Is there plenty of way to get some information on the phi and psi, but not the exact value. The exact value is very difficult to get. But to get which region it is, you can get it also by sequence alignment. You can get it by also deep learning approach, which are giving you some region, but not very precise at the degree pre or even at the 10 degree pre. It's a bit larger than that. And also concerning the chemical shift, actually, it is actually when you do nuclear magnetic resonance, you put your protein in a magnetic field, and then you look at the magnetic moment of your proton, which can align along the magnetic field or parallel or anti-parallel. And then you equilibrate the population of it, and you look how it's coming back to the equilibrium. And that's coming, each small magnet is influenced by the electronic environment around. So it's behaving, it's coming back more differently at the equilibrium because of that. And the chemical shift is more or less the frequency how it's coming back, because it's coming back in a periodic way, so there is a frequency associated. And then when you have something, for example, when you have a proton which is over phenyl ring, you know, like the tryptophan, like the benzene, kind of benzene. You have a benzene, and you have in protein there is no benzene, but you have tryptophan, which have a bit similar, not tryptophan, not. You have the tyrosine, which have a bit similar chemical formula, and you have the proton which is sitting over it. It has a very different chemical shift. So the chemical shifts are related to the environment, and it's in particular some chemical shifts. So the chemical shift of the H alpha and C alpha, they are related to the value of the phi and psi. It was observed experimentally somehow. And it's even so well related that it's possible to build a neural network on which you are entering the chemical shift, and then they are predicting phi and psi.
Okay. So it's chemical shifts you measure for each atom in the sequence?
Yeah.
And you know the sequence?
Actually, you have to do the assignment, but I don't talk about that. You have to actually at the beginning you have a map of everything, but each peak should have a label of an atom. But you can spend several months to try to find who is who. That's
okay, but so it means you have the protein itself, and you measure this chemical shift, but why don't you access directly the geometry? I don't get this, because you have the protein, so you could get the geometry of the protein directly.
I know, but I don't have the protein structure.
So why?
Why? Because usually the protein structures are not coming themselves. I think here is my structure. You have to determine the structure.
Yeah, but you observe the protein, so it has a structure already.
It depends. For example, in the disordered protein, it can populate several conformations. So you don't have a unique folded structure. But sometimes you have a unique folded structure. But even if you have a unique folded structure, you have a unique, a well-defined folded structure, you are measuring other parameters which are related to the distance between hydrogen, and that's helping you to build the structure. Yeah, I didn't discuss that too much actually. It's a bit somehow discussed here actually. No.
But
oui, yeah. Okay, sorry.
No, yeah, go ahead. Sorry.
Attendez, je vais plus du bon, c'est pas grave. J'arrive plus à bouger en fait. Je bouge, mais voilà, je laisse ici. Voilà. Oui, parce que you have something, it's possible. Mais bon, it's a bit yeah, it's possible to build to calculate structure if you know the distance between the residue.
And also a question, because you mostly talk about kind of local constraints, but I assume that in practice, global constraints are important. And if you integrate the thing and you see that you are looping back to a place where you already are, then clearly it's not a plausible configuration. And so,
yeah,
I don't know at which stage.
So global constraints are very important, and especially right now, all the methods of prediction are based on using local constraint and global constraint.
Yes.
But if you have a disordered region, you are losing the global constraint. And if you have not enough big alignment, you are also somehow losing the global constraint. And so this kind of method would have, but okay, now it's I'm not saying I have a very clear idea what to do, but this kind of method of using local information and enumerating could be a way to bypass the global constraint. But then there are trouble because you have some variability of the omega angle, for example, which can be very important. And for the moment, I don't know how to handle that. I'm trying to do something, but it's
And also because, like, how large is the configuration space? Like, basically, how well have you succeeded in narrowing it down? Because at some point you said, okay, 10 at the power 30 states, so clearly that's too much, but 10 at the power 10 would be okay. So I assume that.
So if you have a number of leaves of 10 to the power 10, which I'm getting for fragment around 15 residue, it's okay for parsing the whole tree. With not having too many branches actually, because if you have discreting too much, And then, but then you can get, I don't know, you can get 10,000 solutions, you can get 200,000 solutions. So you have also another thing to filter the solution is to not accept solutions which are too close to the previous one. Actually, you try to get a new solution. Yeah, and that's also and then if I am 200,000 conformation, then I put that in this clustering approach, which is the self-organizing map I didn't discuss much. And then I'm getting, can be between several tens to 100 solutions. And then when I'm doing the assembly step, I am also continuing to cluster the conformation. At the end, I'm typically having 200 conformation when I'm finishing the whole TIPP procedure for an IDP for an IDP protein. So I don't have so many of them at the end. I have been pruning a lot actually. And what I'm finding interesting in this optimal transport approach, I don't know whether it's a bit that I'm a bit dreaming and so on, but it's the idea to try to say, okay, we are in the probability space, so then we keep note that we have such, this one which are there, and maybe you may have a lot of them, but you may imagine you are not, if you know that you have been seeing them, you can always try to recall, yeah, I've seen that. And then the point is maybe you have first maybe to merge also different sets, because if you have two fragments for which you get all the Gaussian probability distribution, then you are attaching the fragment. Some will disappear, but some will be merged somehow. So and maybe then I don't know, the Wasserstein-Barry Center can help. Or I don't know. I mean, it's a bit. But I'm not very. Okay, I don't know much. I've been looking at the course on internet on marine, but not more than that. I mean, I'm not a specialist.
The triangle are local features,
Yeah.
like human practice features. So
Like human.
they are human practice features that people look at how can I describe my protein somehow.
Yeah.
Is there like I don't know a way to infer other way of describing proteins, like with more global features, or typically with a deep learning like encoder.
Okay, there are a lot of, actually, in deep learning, there are a lot of people able to predict the so-called contact map. So you have the coordinate of the here you have the residue number. Here 1 to 100, and here 100. And you put a spot when it's closer than 5 residue, 5 angstrom, I'm sorry, 5 angstrom, and you get, for example, when you have alpha helices, you get something like that a bit. When you have something like that, it means that you have two beta strands. But it's actually when you are doing that, you know that you have some global restraints, and then you are in fact the global restraint, and then you can fold your protein. So that's still the point between global and local information. Yeah? People now they are relying on local and global, because but if you actually what's happening is that if you have variation of the omega angle I've been showing you, which is I told you it's a constant angle, this one. This is the constant angle, this one around 180. Actually, it's a bit. It can be up a deviation of 20 degrees for 180. You know, and or minus. And if you have you look at the protein like a robot arm, which is doing something complicated with a lot of different hinge, then if you have been making some error at the end, you can get different. So that would be another way to describe looking local information to try to see what's happening at the end when for the global fold of the protein. But for the moment, okay, I am trying to look at that, but it's not so easy to predict because it was not so much looking at people.
So maybe another question. So you try to find a map that goes from the world of the supermoon with the different kind of small bricks forming proteins to the geometric configuration, and you surely need to invert the map, like if you want to make a protein on a certain form, so how we will inject DNA. My guess is that you know that the virus has some spike protein, and you want to make something like a cap, surely you want to. How to
modify the mail to make a protein design. But what you said that I was not being so ambitious that I want to solve the full optimal transform.
No, but my point is that okay, what biologists are dreaming about?
Yeah, about design of proteins. They are certainly dreaming about that. Yeah. Actually, David Becker has been able to produce protein, making some geometric form like a triangle and so on, and is designing a protein and then making the structure, and then is actually having a triangle shape. You know, what you told me, having the optimal transport coming from all the set of residue and going to the phi side. I mean, all residue can have all phi side, actually. Most, because the glycine is more mobile than the others, but nevertheless, most residue can populate most phi side. And now there are people working on other kind of alphabet. So that's the amino acid residue is one alphabet for the protein residue, the name of the amino acid. But you can imagine other alphabet. For example, there is a 3DI alphabet for which is inserting actually long range constraints. And then in that way you have the long range constraint which is included in the alphabet. And that may but that would be maybe something maybe a transfer between the 3DI to which is a new alphabet which is used in fold-seq actually. So it was published this year actually. It was around since in bioarchive, which is 1 year, but it was published this spring. And using this kind of alphabet, maybe we could go from the space of this alphabet to the space of phi psi. That's some relation. But with the usual amino acid, I don't know. And of course, if you would have such optimal transport, then yeah, you want to inverse it, then you get the sequence. Consider it, that's for sure. I agree with you.
Okay. Maybe last question, just also about deep learning, like AlphaFold, for example. What inputs they have? They have these angles as inputs, or they?
The AlphaFold is taking the sequence of the protein.
Oh, okay.
It's just taking one sequence.
Okay.
But then it's capitalizing on all the database on the PDB actually. So that's the reason for which actually it's working much better for folded domain than for disordered region.
Okay.
Which for disordered region there are not so many. It's difficult to know what are the structure actually because the people are not determining structure. There are now people starting to predict IDP conformation from sequence by deep learning using all the set of calculated conformation. But it's a bit, you are making a deep learning of something which is predicted. So you don't know which kind, how much you are accumulating the error. Yeah. That's. So AlphaFold is using the database of protein structure, which are mostly folded protein, because you don't have much information for the rest. Because it has several conformation. As it has several conformation, you are getting average, either average in time, either average in space, and then the average is it is averaging to 0. Because most of the method for making structural biology, it's a bit what I was describing here, is it rely on averaging. So the nucleic resonance, you are averaging along the time, and you see what's happening on time, you get an average And the crystallography, you are averaging along the packing of the crystal. So you have a lot of different, and sometimes you don't see, for example, some loop in X-ray structure, because one loop is a bit like that, and the next, the neighbor is a bit like that, and then another position of the loop, and then you are summing everything and you get 0. And then in the X-ray structure, the crystallographer say, yeah, this one we have not seen. And you have the same trouble with cryo-EM also. That's you see, not the same, but similar trouble.
Maybe a question that's more about fully protein that IDP. Is there any influence of changing one or two amino acids in the sequence? Because biologically, I guess this has a huge impact on the structure of the fully protein, but maybe on IDP.
What is the sensitivity of IDP to the mutation?
Yeah.
Alors, okay, for the folded protein, sometimes you make one mutation and it's not folded anymore. You can it's happening, but for most of the time it's not changing much, but sometimes it can be completely.
I guess it's there are beads that are very sensitive if they are in key regions, like the place where the arm will unfold on the hemoglobin.
Okay, I don't know. Yeah, but maybe there's some, I don't know.
Do methods like AlphaFold predict these things well, or don't?
What always it depends. Okay, I think there are people working on that, but I would not say. Yeah, AlphaFold has trouble for a bit sometimes individualizing the protein. But okay, it's a tool which is very powerful. And then if you have one has to cross different tools. But yeah. But concerning the IDP, actually, if you change one residue, it will not change much for the biophysical, but it changes a lot for the interaction with other proteins. And in the cell, the IDPs, they are very versatile, they are very promiscuous, they can interact with many partners, and then the interaction network can be completely disturbed since one residue has been mutated. Yeah.
Because, actually, how do they interact with other proteins? Because I was told the main thing was the structure, like the geometry of the protein to interact with other molecules.
Yeah.
But if there aren't that much of a structure.
And so what's happening is they can interact with folded protein because the folded protein has a geometry, and then they will fit the geometry, and they can, and as they're very flexible, they can talk with one protein on the day and another protein a bit later. It's a bit like.
That's only on the chemical property of the amino acids.
Yeah, it will be. Yeah, it depends on the chemical property of the side chain amino acid, but it somehow depends also on the geometry of the surface of the folded protein. And then you have also the case of two disordered molecule interaction, protein-protein or protein DNA or protein RNA. And there, it can happen very well that they are interacting very well, and both are staying completely disordered. So interacting, but each one having a lot of conformation, each conformation with another of the others, and okay, that has been also observed.
Okay, so we can probably stop there for official questions and then maybe discuss afterwards.